The neuron, also called a nerve cell, is a key component of the nervous system because this is the cell that generates nerve impulses and action potentials. Neurons, which are also known as nerve cells, are specialized cells in the nervous system that are responsible for communication. They communicate by generating nerve impulses, or what are also called action potentials, and releasing neurotransmitters. Neurons have two basic regions. One of those regions is the perikaryon, also called the cell body or the soma, and I don't really care which term you use, long as you're familiar with all of them. The perikaryon is where you find the nucleus. It's the metabolic center of the cell. It's where you find the whole host of organelles that you're familiar with, that you expect to find in any cell of the human body. In terms of structures that you should be able to identify in the perikaryon, I expect you to be able to identify the nucleus and the nucleolus. They're rather large, and if you recall from the tissues lab, you could very easily see the nuclei of the neurons. In terms of organelles, there's really only one organelle that I specifically want you to be able to identify on a neuron in the perikaryon, and that is the nissl substance. Sometimes it's called the nissl body. They are specialized rough endoplasmic reticula that we find in the neuron. So a nissl substance is nothing more than a specialized rough endoplasmic reticulum. The neuron is the basic functional unit of the nervous system. This is the specialized cell which is responsible for communication. And it does this by generating nerve impulses and releasing neurotransmitter. Neurons are also referred to as nerve cells. Nerve cells have two basic regions. The perikaryon, or soma, or cell body, they all mean the same thing, is the central portion of the neuron. It's where you find the nucleus and the nucleolus and the normal host of organelles that we associate with any cell of the human body. Extending from the perikaryon or cell body are the processes. These are fibers which extend from the cell bodies, and these fibers have names based on their functions. In addition to the perikaryon, the other portion of the neuron include the processes. A process is nothing more than a nerve fiber that extends from the cell body. And as you can see in this example, there's several fibers extending from the cell body. The specific name of these fibers really depends on their function. Processes that conduct electrical signals toward the cell bodies are known as dendrites. Notice the black arrows indicating that they are conducting electrical signals toward the cell bodies. Those are the dendrites. Axons are processes that conduct nerve impulses away from the cell body. Notice the black lines indicating that they are conducting nerve impulses away from the cell body. Let's continue to study this axon. It begins at this funnel-shaped area called the axon hillock, and it ends at what are called axon terminals, aptly named. So nerve impulses leave the perikaryon via the axon hillock, move down the axon, and they end in the axon terminals. And in lecture, you'll learn that when those action potentials or nerve impulses reach the axon terminals, the result is the release of neurotransmitter at that axon terminals at a synapse, or where those axon terminals form junctions with receiving neurons. I also want to point out that the axon is covered by a white structure called a myelin sheath. In this example, you have Schwann cells wrapping around the axon, and collectively they make up a structure called the myelin sheath. Also notice that there are spaces between those Schwann cells where the axon is exposed. Those are called nodes of Revere. And in lecture, you'll learn how they function to increase the rate at which a nerve impulse travels down that axon. Okay? Our focus is anatomy, so here the focus is just being able to name the structure. This whole entire white structure collectively formed by all of these individual Schwann cells is called a myelin sheath. The areas of exposed axon in the myelin sheath are called nodes of Revere. You are responsible for being able to identify structures of the neuron on an image such as the one in your book and lab manual, and on these two models. One model, the one shown here on the right, has the perikaryon attached to the axon, and in this other one they are separated, but they're basically showing the same structures. I want to take a moment to relate the image of the neuron in your book and lab manual to these two models. Let's start with the perikaryon or cell body. So here's the perikaryon or cell body or soma on the image, and here it is on one of the models. Here's the nucleus. The nucleolus is this dark area inside. And the only organelle you have to identify are the Nissl substances or Nissl bodies. Remember, they are specialized rough endoplasmic reticulum, although on this model they don't particularly look like rough endoplasmic reticulum, but that's what these little gray structures are. The processes extending from the cell body here are the dendrites. These are the processes that conduct nerve impulses, or I should say electrical signals, toward the soma. Here we have the axon hillock. That's the beginning of the axon, which is this structure here. I mentioned earlier that these white sort of extensions or processes here were the dendrites. And I also mentioned these brown structures here were axon terminals. Let's talk a little bit more about those axon terminals and why these axon terminals forming junctions with these dendrites. Well, at a synapse, which is where a sending neuron comes into close proximity to a receiving neuron, we have the axon terminals of the sending neuron coming very close to the dendrites of the receiving neuron. And so what you're seeing here are the axon terminals of other neurons out there in space. And you can have more than one sending neuron forming junctions with the receiving neuron. In fact, you can have tens of thousands of them. So these axon terminals are axon terminals from other neurons somewhere out there in space. Now let's focus on the axon. So again, here is our axon hillock, which begins our axon. So here is the axon extending here. It's been dissected so you could see the internal components, but this is the axon here. That axon is surrounded by Schwann cells. Here's a Schwann cell here. You can see the nucleus of the Schwann cell here. Here's a second Schwann cell, and here's the nucleus right here. These Schwann cells collectively make up the myelin sheath. And as I noted earlier, we have little gaps between the Schwann cells called nodes of Revere. And likewise, between these Schwann cells, we have nodes of Revere. Now, how will I distinguish Schwann cells versus myelin sheath versus an axon? If I point here and I ask you to identify or name the cell, then I'm asking for the Schwann cell. If, on the other hand, I point here and I ask you to name the white structure, then I'm looking for myelin sheath. If I point here between the Schwann cells and I ask you to identify the space, then I'm asking for node of Revere. So it's going to be very important that you carefully read questions if I'm asking for anything on or around the axon. Oh, and how would I ask for axon? Well, I would specifically ask here for you to name the process, because an axon is a process. So again, if I want Schwann cell, I'll ask you to name the cell. If I want myelin sheath, I'll ask you to name the white structure. If I want axon, I'll specifically ask in this area for you to name the process. Now, I want to get back to the axon terminals that I alluded to earlier. So at the end of the axon, or I should say the axon ends in these little axon terminals here.